It's my pleasure to introduce Dr. Richard Willson from the University of Houston. Dr. Richard Willson is the Huffington Woestemeyer Professor of Chemical and Biomolecular Engineering, Biology and Biochemistry, and Biomedical Engineering at the University of Houston. He is also a member of the Baylor College of Medicine Quantitative Chemical Biology Program, QCB, Computational Biology, the Baylor College of Medicine QCB program, and a senior affiliate of the Houston Methodist Research Institute. He holds a Bachelor of Science and Master of Science in Chemical Engineering from Caltech and completed his PhD in Biochemical Engineering and postdoctoral studies in biochemistry at MIT. And Dr. Willson is a recipient of the Presidential Young Investigator Award under the first Bush administration and an elected fellow of the AIMBE, ACS, and AAAS. He is a member of the National Academy of Inventors and served for a time as the interim Associate Vice President for Technology Transfer at the University of Houston, and then the non-medical public university with the largest patent royalty income in the entire United States. His research interests are in biomolecular recognition, medical diagnostics, and the purification of biological molecules. So, Dr. Willson, the floor is yours.
Thank you very much. Sounds okay? Mike's all right? Good. So I have a lot of stuff to cover. This is a giant topic that could be multiple lectures. And some of the material at the end, I'm going to just put up and say, here's an interesting resource. Here's another interesting resource. Here's another one. You won't be able to write them down that fast, but the PDFs of my slides will be available from the organizers if you're interested in this stuff. So I kept this, I gave a talk at Mount Auburn Hospital last week, I guess it was, virtually. And if you get into this area, you'll have to do this kind of stuff too, right? You will have conflicts of interest. You'll have significant financial conflict of interest, and you'll have to disclose when you give a talk that I have, an IP interest in some of the lateral flow stuff I'm going to talk about, and some of the novel antibody binding proteins I'll mention in passing. So it is a slightly different world than that of pure research and science. So 4 parts. First, my disclaimer. So I'm a regular faculty, right? I mean, I have NIH grants, I have grad students and postdocs and research faculty in a lab. I'm very proud that I have a paper with an impact factor north of 18 about to come out. And we do all of that kind of usual professor stuff. But I was blessed or cursed or marked by a couple of things. First, my father and my son are patent lawyers. And so that was going to happen. But it was not obvious that I would get into this. I mean, I remember being a long time ago, I was on the governing committee that ran the honor system at Caltech. It was a very small community and a very good honor system. And we invented a thing called the Industrial Relations Building, which was kind of spiffy, and they had these green felt pads with nicely sharpened pencils. And I had no freaking clue why there was an Industrial Relations Building for my university. What the heck is that? And later you'll see my naivety continued for many, many years. The later generations of you guys will be better about this than I was at the same age by a long shot. And then we'll talk about patents and this really important and slightly awkward topic of actually having customers. Scientists and engineers are not good at that, and we'll talk about that. And then some practicalities about startups, much of which will be things that you're just going to want to look up on the PDF if you're interested later. So what's a patent? If you teach society something useful, they will let you be the only practitioner for a while. It's a deal. That's the trade, right? You are rewarded for giving away your valuable new invention, your secret, to society and teaching everybody, teaching them well, enabling them to use it with a delay before they actually start doing it. And the idea is that science and technology will build upon what you've disclosed. They'll make better things after the term of 20 years. Everybody can use it. Society can advance. So it is only a right to exclude. It doesn't mean you can practice your invention. So if you get a wonderful, strong patent on green cars, for example, but somebody else already has a patent on all cars, then they can't do green cars, but you can't do any cars. So your patent can be dominated by one that's broader or reads over your area, okay? So there's a different thing that comes up called freedom to operate, which is if you want to practice your invention or do whatever it is, you need to make sure that nobody else has got patents that bar you. I will note that In several startups that I've known a lot about, patents were most of the value. They really were a very substantial creator of value in a startup, in an M&A situation, for example. So suppose you have a patentable invention. What do? Well, first, I'll show you some patentability criteria you should think about briefly. You should think about who the inventors are, right? Inventorship is a litigatable issue in fighting over patents. So you have to get it right, or the patent might be invalidated completely, which would be bad. Don't disclose it publicly. In general, you can blow patent rights really quickly by giving a public disclosure of your technology, whatever it is you invented. And go and talk to your tech transfer office. And I'll list a few of them on a slide in a minute. But every institution represented here surely has a tech transfer office. There's actually some federal law that sort of drives that. And it isn't anymore, and maybe none of you thinks that this is the way it's done, but my generation was used to the idea of, oh, I've made an invention, I write it down in my lab notebook very carefully, and I get my friend to witness it. And it would say, witnessed and understood by Bob Smith on this date. And that marked me as the date of invention, and if I was the first to invent, I could get the patent. It doesn't work that way anymore. By harmonizing with the rest of the world, under a thing called AIA, the America Invents Act, we no longer have first to invent, we have first to file. And first to file is a recipe for being quick to get to the patent office. And so that means you should call your tech transfer office up, email your tech transfer office, go to their website promptly. And they'll be happy to hear from you. I used to oversee our tech transfer office, and we're always happy to hear from inventors. And it would be rare for us to say it was too early, frankly. And we would never scold anybody for approaching us. So to be patentable, it's got to be new. It has to be useful for something, although it's usually pretty easy to think of some utility for almost anything. It has to be non-obvious. So this is a place where you'll fight when trying to get a patent. The patent examiner will say, I see your green car is interesting, but you know, green is already known and cars are already known and combining them is obvious. You'll get things of that structure. They'll take two references, and then your job will be to say, no, there's all these reasons those two things are not naturally combinable. The prior art teaches away from combining them. Our discovery of combining them gives unexpected value, and you'll fight back and forth in the examiner in a way that we'll talk about in a minute. And then subject matter. You can't patent mental processes. Algorithms are tough, although there's some narrow paths to go there. You can't patent things you don't enable. So it would be very difficult, for example, to patent antibodies that bind to insulin. You could patent some particular antibody that binds to insulin, but patenting all of them, especially under a new decision called the Amgen decision, it's very difficult to patent a very broad thing. And products of nature. So you might be able to patent erythropoietin in its purified form because it doesn't appear that way in nature. But patenting it sort of in a general sense, you can't. You have to enable the teachings. That is, the societal deal is you give society the knowledge and the ability to use your invention, and in exchange, you get the monopoly. But you don't have to have done it all. And this is called prophecy. Now, writing a prophetic journal paper is called academic dishonesty and gets you, blown out of the career, right? You can't do that. But in patenting, a certain amount of prophecy is just fine. I personally have had a significant number of issued US patents that were entirely prophetic, where we filed a great big application. It got split up in a process called divisional, and we had done some of it, that issued. Other things had not been done. Those issued too. I think all of them were then later done by others showing our prophecy was correct, but we owned it before they did it. So don't be too shy about that kind of stuff. You owe it to yourself and to some extent to your tech transfer office to look and see what's out there before. And I also want to emphasize that, we all read the journal papers, see what's going on in our field. We all ought to be reading the patent literature too. And there are classifications, like each little area of science and technology, patentable material, has a classification, a subclass. And if you find the right class for your stuff, you will find a lot of the leading edge work in your field, which isn't in journals. It isn't in journals, it's there. So we're making some better practice now of following patent literature areas we care about. Also, if you're going to file a patent, you really owe it to yourself to see what's out there. First, to make sure you're not just completely scooped and it's a worthless exercise and you should save your time, but also, you know, you can strengthen your application by avoiding what's out there previously and say, oh, but we do this and this is not in the prior art. You don't own your inventions. Essentially, all of us, as a standard condition of our employment, have to assign our intellectual property to our employers. Right? All of the members of the Gulf Coast Consortium, for example, I'm certain have a policy like that. The failure to sign the form probably will not get you around that. So your institution owns the intellectual property. There are laws. There's a thing called the Bayh-Dole Act, which we could get into. But they will give you a cut of the net profits. At U of H, we will give inventors 40% of the net profits of an invention. I emphasize it's not the total royalties. It's certainly not sales. It's the net after our expenses for patenting. Now I'm speaking as if I were the tech transfer office. That can be great. We had a single inventor who made probably by the end more than 100 million personal dollars. Right? So it can be a pretty good outcome. Everybody perks up. I once said to some people, the way to get more disclosures into the tech transfer office is to have more Lamborghinis in the parking lots. So yeah, it's very highly incentivizing if it works out. You need to go and talk to your tech transfer office. I listed a bunch of them here. It'll be on the PDFs, but this is, you know, a 10-second Google search for each of you. Just find your tech transfer office. And again, they'll be happy to hear from you. You're going to think they're bureaucratic. They have a hard job, right? I'll tell you now that most people are not entirely satisfied with their tech transfer offices, and that's not entirely the fault of the tech transfer office. They have this issue that they have all kinds of constraints, both legal, financial. They're besieged by ideas from lots of people sometimes. And one of the big problems is, you know, at U of H, we have, say, 1,500 faculty. We have maybe, you know, 5 or 7 or 10 people who are thinking about all the inventions that come out of them. And 1,500 to 10 is 150 to 1. So nobody can fully understand all of the work of that many other people, especially the most inventive stuff, which covers, you know, the leading edge in a bunch of different fields. So they're not going to be super experts on your topic. You'll have to educate them. The other thing, and now I speak as the guy who used to sort of defend my tech transfer office staff from the faculty and the students, don't expect to just sort of toss them an idea and get back a great deal of money. It's a cooperative effort. You have to do the work. I'm sure you're willing to do it, but I just want to adjust your expectations a little. Success really does depend on the inventor. And there are 2 big places that inventors have to play. One of them is it's easy to write a very weak patent application which will not have any actual value in the long term. We'll talk about that in a minute. And the other is that the inventor is the person most likely to connect the tech transfer office to the person who pays for the invention. That is, if you're a super expert in your little field, and you know the players, you're good enough to make leading commercially valuable inventions in that space, you are very likely the person who can point to something, oh yeah, and by the way, I know Merck is interested in this area, talk to this guy at Merck. That's the way. I read a statistic, I don't know if it's true or not, that 80% of such licenses are done. Been my case. But be nice to them, right? They are aligned on the goal of making you a lot of money. So you should like them and value them and support them and don't be mean at them and don't yell at them. I had to yell at a few faculty occasionally, tell them to be nice to my staff. Now, everything I said up to date assumes that you are working together to develop intellectual property that will get licensed outside the system, say to Merck, for example. Now, you might want to do a startup, and we'll get to that. Then it's a little more complicated, right? Because you don't have an adversarial relationship, but you have a negotiating relationship with them, and they will want some stuff. And while they want you to succeed, they also want to get their cut. When you approach, there will be a form. They're going to want to know who the inventors are. They'll probably want to know the credit split. So make sure you're reasonably comfortable with that. You know, it isn't good when the inventors come and disagree over the credit split once they're interacting with the tech transfer office. Are there inventors from other institutions, right? We are a good example of interinstitutional collaboration. It's fine. That can be managed. Most of our institutions around here already have arrangements for handling that sort of thing. but they'll want to know. They'll want to know something about the invention. They'll be quite interested in the market, right? Drugs are good. They tend to sell lots of dollars. It costs money and effort to get patents. And so getting a patent that addresses only a tiny market may be good for your ego, might establish a certain stake in the ground for your intellectual priority, might not be financially worth it, right? So this will be an issue. And they will be deeply paranoid about any plans you have publicly disclose this. Right? It's grotesquely unfair to them to come in and say, hey, I'm giving a talk at ACS next week. Here's my idea. Don't be that person. Don't do that. It's really mean to them. They will probably scramble and do what they can, but the quality of your coverage will be pretty bad. And it's not a question of weeks of notice. Give them months, because you want them to have time to find the right attorney to work with you. And I'll talk in a minute about the writing of the application is a pretty big deal. I'll note that usually they start with a provisional application. Those, if done well, are not extremely cheap, but it's the normal thing. It adjusts sometimes. It gives you a year. It's not examined at first. It will die quietly if it's not converted, but it does hold your priority date. And I'll tell you a trick. At U of H, and I'm now speaking from my standpoint as an inventor, we provisional almost everything. Practically everything gets out for provisional. And then after the year, we have a committee and we have the staff and we have outside advisors who help us decide whether to spend the money to convert the provisional to so-called non-provisional application, which starts the examination process, means we're spending some money. And we don't convert them all, right? I would guess that most institutions convert about half, let's say, with a big range of variation. So what happens if the committee decides not to convert your provisional application into a non-provisional? Well, U of H will give it back to you. We'll give it back to the inventors. We get a little slice if somebody makes a lot of money, but basically we give back control. But it also means we give back the costs, because we, the office, we're paying the attorneys to do the provisional, but if we're not going further, you, the inventors, are now paying. So if you're an inventor, where do you want to get as much work done as possible? By expensive attorneys? At the provisional stage. So that when they give it back to you, it's a highly mature, nice document, which won't cost you too much to mature to the point of a good, strong non-provisional, right? So it's in your interest, even if only for that reason, to work strongly on the provisionals and do a good job. It also is vitally important in competitive fields, because in that year, somebody else may come in, and you don't want to have to like add a bunch of new material after a year has passed just because you were lazy at the beginning. You might well get scooped on something important. Okay, so I said there are two places you got to work hard. Drafting a good patent application should hurt. It should be a difficult and painful activity. It's, you know, it depends a bit on your sort of the fluency of your imagination and stuff. A couple of my grad students probably have traumatic memories of writing provisional applications with me. The problem is, if you write, you know, an extremely narrow patent application, that's all you're going to get. You won't get more. You can only get what's in the document, what's in the specification, what's in the examples. And that's not very valuable, right? I mean, my father had this wonderful phrase, the coverage of that patent is about as broad as the thickness of the paper it's written on. Worthless, a net zero or net negative, because you just wasted money to write it. And so when you are, for example, thinking about some interaction where there's binding, right? Maybe you're doing a sandwich immunoassay, maybe you're doing cytometry, maybe you're purifying something, maybe it's a drug, you know, explicitly list antibody, SCFV, aptamer, lectin, receptor, substrate analog, all the things you could use for binding. Because you're going to end up searching through the specification for words to support some argument or claim modification that the examiner is going to be driving you to make. And you want to have those words in there explicitly in black and white. My advice is give till it hurts and then do it again later when you're fresh. Right? You can't do it in one session. It's not an all-nighter kind of thing. You have to go away, refresh your brain, be exposed to other things, have new thoughts, write them down casually. I keep a to-do list on my phone. I keep updating and updating over, you know, it could be weeks, many sessions. If you can write 20 distinct examples that embody different forms of your invention, that's pretty good. You might end up with a bunch of different patents as a result of this. And you're the only one who can do it, right? Your tech transfer office doesn't know the technology well enough. The attorney doesn't know the technology well enough and shouldn't be an inventor, right? So you've got to do it. And this is a place where, you know, it can be the highest dollars per hour labor you ever do, even though it can be kind of painful. Yeah, so the last line is my reminder to tell you. So some of you will know, how many of you know what staphylococcal protein A is? You ever purify antibodies, or you know about Staph aureus? Not so much. So antibodies are a $200 billion a year business, pharmaceutical monoclonals. They are essentially all purified using a cell surface protein from Staph aureus, which likes to bind to antibodies. It's a virulence factor for the bacterium. It also binds to von Willebrand factor and forms biofilms and induces apoptosis in some of your immune cells. But it's a very useful tool for the affinity separation of antibodies out of cell culture media. Somebody grows a giant tank full of Chinese hamster ovary cells. Antibodies are 10 grams per liter. You want to purify this stuff. You spin the cells out. Maybe you filter. You run it over that column. The protein A grabs the antibody. Okay? It's a big business. We had the thought that the pioneers in this field found, quote, protein A, Staphylococcus protein A, from one strain of Staphylococcus aureus. And we imagined that maybe there were others. So we now have 960, some of which bind as well as the canonical protein A, and are not under the existing patents for protein A. And in fact, there are patents on ways of using protein A. Some of our things are probably not really protein A by the definitions in those patents. So we can practice all of those other inventions by using our non-protein A binders. The provisional was 130 pages long. My student was ready to murder me by the time we filed it, but I am rubbing my hands with glee, right? That's the situation you want to be in, tired but happy and confident. Here's what it looks like when you file. You send it in and it gets examined. The examiner will look at it. And then it goes down to the decision point in a little flow chart here. And there's the left path allowance of all claims. If you get that on the first try, which I did once, and I was bragging about it to somebody more experienced than me, what he said was, oh, you didn't claim broadly enough. And he was right. I hadn't claimed big enough, right? It's like the old story about using the whole tennis court. You know, you want to sometimes hit out, and I hadn't used the whole range. But you won't. Examiners will practically always, you know, bounce things back for some reason. And then you'll make amendments and requests for continuing exam appeals, all kinds of stuff. And you'll go around and around a few times. It's not unusual to go around that loop on the right, you know, 3 or 5 times before you get a patent issue. If you really care a lot about this, the patent examiners have to follow a rule book, which is here. Don't try to quickly write it down. It will be in the PDF. Patents have very specific parts. There's a standard format. I gave a paper to ChatGPT-4 and asked it to write me a US patent application in standard format from that paper. And it wasn't bad. It wasn't bad. You know, I wouldn't trust it. It might hallucinate. But in terms of getting all the forms correct, the formats correct, it was pretty good. The drawings have to be a special format. There are online tools for that. The specification is that technical description where you've got to be long and verbose and give a zillion examples because you'll be forced to sort of find patentable material in there. And then the claims, which I have in bold, literally every word matters. There's a lot of litigation over patents. And while I'm not a patent lawyer, I do a fair amount of legal expert witness stuff. I was expert in the first so-called biosimilars case to go to trial in the US. I was an expert in the one that went to the Supreme Court. And so I've been exposed to this process pretty extensively now. And I have actually seen people fight over the meaning of the word the. What is the protein exactly here? So pros in this space always turn to the claims first. It doesn't really matter what the title is. It doesn't really matter what the abstract is. Things may have mutated an awful lot when you went around that iteration cycle of modification 5 times. What really matters is the issued claims and exactly how they're worded. And sometimes the word in the claims has been defined somewhere in the patent in an unusual way. So if you're interested in a word, look for it. Sometimes there's a section of the patent that says definitions. You can find it. There's a tradition of inventors being, quote, their own lexicographers. Patents have to be public because they state a claim to an area, right? So everybody has to know what the patents are, see what's claimed. And you can find them here. Google Patents is good, but some of these others are pretty good too. There is a classification system. I recommend that if you find a patent that looks like your research or your interests, find the classification and look for other patents in that classification area. You may find some very, very interesting stuff that way. I'll tell you a personal example. This is old. It's from days when the U of H patent system was way less mature than it has become, way before we had some really valuable patents. So this is an infrared camera image. This is from a time when infrared cameras were expensive too. And it's showing transformers up on a power line. And the one that's glowing is overheating, right? There's something wrong with it. You could do this for insulation on houses, et cetera, et cetera. These became much more available during the Reagan Strategic Defense Initiative called Star Wars, where there was a lot of interest in detecting warm missile warheads coming at us against the cold of space. There's a great deal of technical progress in that space. This was early mid-90s kind of. So one of my appointments, my main appointment, is in chemical and biomolecular engineering. And while I'm pretty biomolecular, some of my colleagues are real chemical engineers. And they make catalysts. You don't need to know much about catalysts. Just suffice to say that, you know, all of those industries in the ship channel, the vast multi-hundred-billion-dollar industry that's east of us, practically all of it runs on catalysts, which will polymerize plastics, destroy waste, convert crude oil into gasoline, et cetera, et cetera, et cetera. So catalyst development is a big deal, and a good catalyst can enable the economics of a process. The problem is you don't know what to use for the catalyst. And so I have this, you know, billions of possible combinations. You could get some alumina, right, a ceramic, and put copper or iron, or a 90% copper, 10% iron mix, or 90% copper and 10% nickel, or 90% copper and 10% manganese, or copper and iron, or iron and nickel, or tin and copper. It's endless, right? And so the classic way of testing these was to make a batch, put it in a little reactor, set it up, let it equilibrate, sample the products, and then analyze it in GC or HPLC and see how it was doing, and then go into another one. And, you know, PhD chemical engineer could do like 10 combinations in a day. So I was very interested in combinatorial library screening quite early. I'm pretty sure I got the first NSF engineering grant for phage display. I doubt I got the first NSF grant for phage display, but it was early. It was in the early 90s with a guy named Mike Benedict then at A&M. And so I was thinking a lot about library screening, but I'm walking around, a bunch of my colleagues are doing catalyst development. And one of them was looking at reaction wave patterns with an infrared camera. Well, most reactions liberate heat. Maybe we could look at a collection of catalyst pellets with different metals on them in a single reactor and just see the active ones light up. This turns out to work out really well. Okay, this picture is not from my paper, it's a newer one, but you know, these guys are active and these others are not. You can they just light up like stars. And so this works really well. And I disclosed it to the tech transfer office, and I got back and this is not very highly developed at that time. I got back basically a rejection. Said, well, the committee said, we don't see the value. Maybe it's useful in biotech. No, you idiots, you completely missed the point. Right? Didn't say that. And I went to the person in charge of the tech transfer office, who'd been seconded to us from a federal agency that was known for being pretty conservative about patents. It's a long time ago. And I said, well, you know, my name is actually Richard Cole Willson III. My father is Richard Cole Willson Jr. I've mostly forgiven him for giving me this name. He's a patent lawyer. We've always wanted to do a patent together. It'd be kind of fun. And could he be the attorney who, you know, wrote the patent for this thing, if you guys don't want it? And she said, well, only if he had licensed it. And honest to God, I said, licensed it? What is that? I did later become in charge of all licensing. And she opened a drawer and she pulled out some paper and she, honest to God, brushed dust and said, here's some boilerplate we got from Harvard. Send this to him and see what he thinks. So my father's done hundreds of millions of dollars of patent licensing in, you know, hotel bars in the Middle East. And I faxed it to him, showing when this was. And he sort of laughed at the archaic language and wrote a small check to the university, and we're in business. So, fine. We did a demo that looks a lot like this, and we sent it to Jax. And Jax said, not interested. passed without external review. Quote, chemists are not interested in this area. Okay, fine. That was in December of 96. Yeah, 96. Crickets, nothing happened for a year and a half. But the summer after the summer, 18 months later, it was cited 3 times in Science and Nature, big Science and big Nature, not the derivatives. And the world went nuts. Combinatorial materials, combinatorial catalysis, crazy. The paper's now been cited 320 times in a journal that doesn't normally get that many citations. I was called in front of 500 people, the father of combinatorial catalysis. We continued prosecuting the patent. It got split into a large number of patents because I had millions of examples and the examiner required us to split them out. We did a little bidding war to see who would be interested in it. We talked to giant chemical company this and huge petroleum company that. We at one point were the only technology ever licensed in by SRI, the Stanford Research Institute. We started a company with them. They started paying me what I thought was a crazy amount of consulting money. We went to the Department of Commerce NIST ATP competition, and they awarded us $2 million. And then there was a glitch because we'd back contracted some of the work to the SRI labs, and there was some issue in the legal contracting. This was late 90s. And okay, well, see what you can do. We can't really fix that. Oh, what are we going to do? Start. And SRI had been very careful. All the documents were marked for negotiation purposes only, not legally binding, not settled, not final. Well, we'll go over, we're at Stanford Research Institute. We'll go over to the venture capital. It's on Sand Hill Road, and we'll get the money from them. Okay, reasonable. It's a pretty good prospect. So, I didn't go on those trips. I did later go to Sand Hill Road for something different with Jim Briggs' company, actually. How about that? I'm going to leave that story for Jim mostly. But they went over to Sand Hill Road, late 90s. Most of you won't remember this, but you may know there was this internet bubble, right? So they would walk in and say, well, CombiCat, as we called it, that was our company, had sales projections that looked like this. We'll all make a lot of money in a few years. But the people just leaving the office would be like pets.com, which had sales projections like that. We'll make a vast money in a few days. And while they may have been wrong, the venture capitalists believed them and didn't believe us. So they couldn't raise the money. And we noted that all of the documents were carefully stated to be not binding. So we pulled the IP and sold the IP to another company that had just gone public in the mania of the stock market at that time. And it was a good outcome. We eventually got, this is the base patent 606.363, the only patent number I know offhand for 20 years now. Most patents are not cited by any other patents. Pretty successful to be cited by 5. This one is cited by 200. So it was a very good outcome. I'll tell you how we're for time. I'm just going to tell you briefly that in my PDFs, you will find a bunch of stuff about the CRISPR patents. And I'm not going to go through it in detail now, but you may know that Doudna and the Berkeley folks didn't get the main patent. They got the Nobel Prize, but they didn't get the main patent. And this is the story of the MIT patent that sort of defeated them. And the reason I wanted to put it up is that the so-called prosecution history is public. It's new enough that you can go to the web. This is the patent. Feng Zhang is the sole inventor. You'll see the application reference. So yeah, right. If you want to see the whole prosecution history, all the exchange of the examiners for any modern patent, this is of course a famous one, but anything else you're interested in, you can find it. Just go to the website that's here, Patent Center, and put in the application number, not the patent number, because an application is unique. It may produce more than one patent, and you can find all kinds of stuff. So this is what happened. They filed this thing. They got a typical first alphas action. Everything was rejected. 1 through 20 are pending. 1 through 20 are rejected. There were some formalities. The examiner thought it was not well enough written. They had to be edited a little bit. Then there's some substantive arguments, and they said, well, it's obvious over the combination of these other previous papers, Sontheimer and Makarova. And then the examiner says that it's obvious to one of the ordinary skill in the art. The inventor, Feng Zhang himself, makes a declaration saying, no, really, these things don't go together. I'm declaring that, in fact, it's not obvious to combine those two things. These are different types of CRISPR-Cas system. They don't go together. And then his attorneys called the examiner and did an interview, which is a pretty common process. You, as an inventor, may participate in an interview. Then they modified the claims a little bit to try to satisfy the examiner. And then they got rejected again based on the Berkeley work. And then there was a huge fight and, you know, et cetera, et cetera. Next step, licensing. So you get your patents, right? Or you have a reasonably strong IP position. You're hoping to get patents. You want to get some money, right? So I leave aside the possibility that you're starting a startup. That's next. But now there's the idea of going to license to Merck, right? I know that Merck will want this thing. So again, the tech transfer office needs your help. They don't really know who to license it to. Right? They're not deep experts in all of the fields of all the things going on. And so they need your guidance. So you got to help them. There are a bunch of different ways to do licensing. So for a drug, it's commonly an exclusive license because somebody's going to spend a lot of money on clinical trials and they don't want anybody else being able to sell the thing. But for example, if you invent recombinant DNA, the so-called Cohn-Boyer patents, then you can just do license to everybody. Yeah, give us $5,000 and you can do recombinant DNA. And Cohn-Boyer did that. Or there can be field of use licenses, like, you know, you can license to one group for some clinical application and one for some laboratory non-clinical application. And neither of them cares that you sold the thing twice because their interests don't overlap. I sold something once, or at least came close to selling it this way, with a small scale lab thing for nucleic acid purification and a big industrial scale purification, you know, where Omega might have been the first one and somebody like Merck for DNA vaccines might have been the second one, or human use and veterinary use, right? So don't overlook the possibility that you may have multiple customers who will let you sell to each of them and they won't care that you're getting money from both. You can get royalties. There can be big royalties. I mentioned that one of the Vimpat inventor, I think our U of H would probably run about $400 million in royalties out of Vimpat. Gatorade made billionaires, remarkably. People sometimes think they're going to get, you know, 70% of net sales. It's never that way, okay? 4% is a common number. This is a big expectation adjustment moment, right? 4% of net sales. But notice that if you got two inventors, and the inventor shares 40%, and sales are $1 million, each inventor gets $8,000. Now, I was eating government cheese. I was poor enough as a grad student to, I would really have valued $8,000. But in the scale of life-changing things for your great invention, you really want to aim for a bigger market. Right? You really have to sell a lot of stuff to make a lot of money out of royalties. Okay. Then there's the question, should you do a startup? How do you decide? What do you do? This is a book written by a guy I took a class from once. U of H has the #1 undergraduate entrepreneurship program in the United States. And Rice has the #1 graduate entrepreneurship program in the United States, according to Princeton Review for the past several years. So we live in a charmed environment. And I just love the title. This guy was fun to listen to too. So you have this great idea in intellectual property position. How do you get paid, right? So this is a really important part of this talk, which some of you are going to think, well, he was pretty good at the beginning and then he went sort of wonky. This is the important part. If you want to get a bunch of money out of somebody, you have to meet some need that they have and that they know that they have. And you, assuming you're the typical STEM people that I would expect to be in this audience, have no particular reason to be either trained or good at that. Okay? So the customers have things like pain points, and there's a term job to be done. Granting agencies, you know, the NIH wants health impact, NSF wants novelty. Investors want return on investment, but they also, some investors have so much money that they're playing with this for fun, and they mainly want a cool story to tell at parties. There really are investors who are like that. Or they're giving back, right? So the Rice Business Plan Competition is a phenomenal event that's going to be held here fairly soon. And there are 2 different investor clubs that invest in those companies. And the average net worth of the more rich of those 2 groups is probably north of $700 million. So when that group puts a total among all of them of 800K into some company and transforms the lives of the founders, it's round off air for them, right? It's mainly giving back to the institution. Oh yeah, so here I'm telling you, you've got to be really careful to understand the needs of the customers and investors, and I'll talk more about that in a minute. But sometimes you just get lucky. So NASA was planning a 1,000-day Mars mission a few years ago. Now they've sort of gone to the moon and they'll get back to Mars later. NASA Johnson Space Center is the NASA center that worries about keeping the astronauts alive and healthy. And they wanted diagnostics. So if somebody got sick or some fungus grows on the instrument panel or starts eating the plants that are keeping the astronauts alive, they want to figure out what that is. And you can't bring an infinite number of monoclonals, so you're probably going to do molecular, DNA or RNA-based identification. There also was a bit of a moment for DNA vaccines at that time. Merck had a HIV vaccine candidate, which was adenovirus plus plasmid. They were building a factory to make lots of plasmid. And so there was a lot of interest in purifying nucleic acids, especially in zero gravity. The old phenol chloroform thing, a blob of phenol chloroform floating around in zero G, ready to dissolve your eye is a really frightening thing. So they really needed some new technology. And so we got some NASA money to do some work in that space. Okay, so we did some research. We developed some new methods of purifying nucleic acids. There weren't very many engineers who thought about this. So we were sort of, in the open blue ocean field, if you like, and we published some stuff. And then the Merck vaccine candidate crashed and NASA wasn't going to Mars right away. And so crickets for a while, nothing much happened. And I get this email, little company in Boston. And they say, hey, we're interested in purifying nucleic acids. Can you come out and tell us how to do that? Okay, sure. So I got there. 2012. There are 15 people. They're in a second floor sort of walk up, near MIT. And they're very small. And I give them this talk. There's an audience. They have, as I say, 15 employees. So if there were 12 of them there, so it was Moderna. It's Moderna. So several of my audience are billionaires now. I became a founding member of their technical advisory board, and it was all great in lots of different ways. But if you ever make a bad financial decision, remember, you met the guy who, before the COVID pandemic, had been working with Moderna for a long time. They'd abolished their technical advisory board at the time of the IPO. All the analyst predictions were, it's not really going to work really well. I'm not so excited. So I took some money off the table. I sold a bunch of my Moderna shares at 17. Oh, 17. Fortunately, I didn't sell them all. So it was a great outcome. It was the best consulting gig I ever had. It's a bragging story, kind of fun. But, I was lucky because I had what somebody wanted and they found me. You can't rely on that. You have to go out and find them. And so this issue of identifying needs, what's called customer discovery and product market fit, is the major gap in the tech transfer and entrepreneurship activities of pretty much every sort of STEM graduate student, if you like, and postdoc and faculty member. You probably read the first two quotations, right? It ain't necessarily so. They will not come and find you. So the problem is that this activity, finding the genuine need, is called marketing. And technical people think that marketing is beneath their dignity, right? They think, well, what's that? Right. But in fact, it's critical. And it is a thing with skill and technical knowledge and a whole body of techniques and methods, which the typical science and technology person doesn't appreciate and is actually really bad at. There's the old joke about the gregarious engineer is the one who looks at your shoes while they're talking to you. Well, that's not somebody who's going to get to know the customer really well and identify their needs. So another way to put it is, we all test our scientific hypotheses very carefully, right? Our papers are models of logical structure. We know this because of this, et cetera, et cetera. But our marketing ideas about who will want the thing we're doing are just guesses, half-assed guesses, very poorly supported. And they're not tested in any serious way. So there is a whole way of doing this. The I-Core, I see I'm getting short on time, so I'm gonna be quick now. The I-Core will send you to talk to 100 potential customers. One of their great themes is get out of the building. You will run out of friends who will say your baby is very beautiful. You'll start getting honest feedback. You'll be sad. You'll probably pivot. But if with any luck, you will pivot to a much better target where your product is actually aligned with the customers. Instead of living in blissful ignorance and saying, oh yeah, everybody's gonna buy my thing for this. We did this with a technology I may get a chance to talk about. A guy named Steve Blank is the great guru in this space. The federal government comes and says, we're from the federal government. We're here to tell you how to be innovative entrepreneurs. Most people would sort of laugh at that phrase. they were clever enough to hire Steve Blank, who's actually very good at this. And you can look him up, and there's a bunch of free content on it. Company formation is horrible and painful and takes a lot of money. I'll call out a thing called the E-myth. Actually, a Baylor faculty member was the one who introduced me to this idea. I thought she was very sophisticated about her company things. Oh, I've read the E-myth. I had read the E-myth. I didn't know what it was. The E-myth is the idea that if you're good at the technical work of a business, that you will be good at running a company that does that business. So, for example, that a physician would naturally be a good manager of a hospital, or engineers would be naturally good managers of engineering firms. Drug developers would be natural CEOs of drug companies. None of those statements is true, right? These are different skills. And so even if you're a brilliant drug discoverer, don't assume you're particularly good at running a drug discovery company. This is the E-myth, all right? But you can take advice. If you want to have a depressing afternoon, read this book. It's even ugly. I mean, it's a horrible yellow color. It's not very well printed. But what it does is it pokes holes in the eagerness to file patents. Because people do it for pride, they do it because their institution wants more patents, et cetera, et cetera. Most filed patents don't return their costs and certainly shouldn't be the basis of a company. So if you can get through this book and still be comfortable and confident with your invention, that's a big step forward. And I promise you, it's better to be slightly disappointed early than to invest a year of your life and all of your money in something that doesn't work out. I'll reiterate, starting a company is a business activity, not a technical activity. We in this room are selected for technical ability, not business ability, but there are millions of resources, which I'm now going to flash through. You can look at the PDFs later. So a very good book called Business Model Generation, a thing called the Business Model Canvas that will make you think about distribution channels, customer relationships, suppliers, all the stuff that we normally don't think about. There are a zillion free newsletters and podcasts. Med City News is surprisingly good, actually, for us. And the one at the bottom, the Innovation Map Houston Innovators thing. There are a zillion accelerators. Accelerators are great. These are groups of people who will take you in some fixed period of time, commonly like a summer or something, and they will help build your startup with all of that stuff that you probably haven't been trained to do. TMC Innovation does a bunch of these things. I'm just going to run through these. There's one for cancer therapeutics in particular, partly super funded. Red Labs, Rice Allspark. If you're from Rice, their applications for this summer close in 12 days. If you're interested, I would urge you to do that. BioWell, if you're interested in synthetic biology, I was at a meeting a couple days ago. They're planning to establish a 2,000-liter pilot scale fermentation facility for SynBio stuff on our campus over at the Schlumberger place that we bought. We have a lot of big incubator and company housing space. One of my roles as AVP was to launch that building and bring in the first clients, first staff, and it's grown out pretty well. This thing will be actually be in a big warehouse that's across the street from that. I'm sure I've missed a bunch, but the Canon UH Small Business Development Center, they'll help somebody with a donut shop, but they'll also help you with your tech-based startup. Fanon Innovation Studio, if you've got a great technology, you don't really think you want to do the whole company, go to Fanon, they'll license it in, you work with them, and you can share in the profits. How do you pay for it? So rule one, don't spend money, right? Stay non-launched as late as you can, plan to live cheap and sleep on somebody's couch. These are things I've actually heard from people. Every one of these is an example, actually, that some friend of mine or former student of mine has done. The money most commonly comes from SBIR and STTR grants, and you guys can learn to do that, and you can get a lot of money. You can get millions of dollars this way. Non-dilutive, they don't take any shares. There's an April 5 event in Houston if you're interested in this stuff. I should rep the Southwest Pediatric Device Consortium. I'm a member of the governance of that. We help accelerate the development of medical devices for children defined as everybody under the age of 22. The FDA funds it because there aren't enough of those. We just got renewed for another 5 years. We also just absorbed Minnesota, which is a great medical device area. And CPRIT has all kinds of product development stuff that they want to fund, all kinds of them with big money too. Business plan competitions. If you are a student of any sort, and I'll tell you, I once watched a very clever person sign up for a part-time law school, thereby become a student, and thereby be eligible for one of the competitions and get a lot of money that way. So a certain shrewdness is rewarded. Rice is the best, biggest, richest business plan competition in the world. It's coming up in about a month. If you can attend, I recommend that. Very interesting. Texas A&M New Ventures. The Houston Angel Network, the biggest network of angel investors in the United States, is here in Houston. This is a bunch of stuff they do just to sort of illustrate their activity level. You know, rich people pay $2,000 to be members, and then they invest money in little companies. One of a spinoff that I may get a chance to talk about briefly got funded that way. Venture capital is hard. You'll be way down the road if you get to that. But I highly recommend this book called Venture Deals by Brad Feld. Where to put it? Stay in your own lab, be at your house, don't spend money, don't pay rent, or look at it, or live in a corner of another company. A friend of mine did this. He bought the beer on Fridays. They let him have like one little tiny bench. He had no rent. He could use their equipment. It was great. And then there are all these other places where you can be at U of H, at J Labs. QBio is new to me. I learned it when I was setting up for this thing. U of H has 250 lab benches and 28 fume hoods. It's a fairly big enterprise. Okay, so should I stop and take questions, or should I talk for another minute?
Questions, anyone? Thank you, Dr. Willson. I'll ask one question.
Sure.
The FDA has a new regulatory pathway called live biotherapeutic products. Could you talk a little more about if you're able to patent a non-wild type organism versus maybe a genetically modified organism for the purpose of this new regulatory pathway in the FDA called live LBPs, which are basically probiotics that the FDA has finally taken under their wing?
Sure. So the short summary is that the question is well beyond my limits of my knowledge, but I can say a few things anyway. We have our pediatric device consortium often gives money to companies to engage with a regulatory advising group called Proxima. And so there are people who specialize in this. Certainly, living microorganisms are patentable. The Ananda Chakraborty first living organism patent is now probably 30 or 40 years old. So patenting a novel strain of a bacterium is certainly possible. But the finer points of your question I'm beyond. I think your mic isn't working. Yeah, just yell, it's fine.
Okay. If you have suggestions for resources that are the intersection of biology and intellectual property, like specifically if you wanted to patent something that is biologically related, where do you go to learn about that kind of stuff?
It's a good question. My experience has been that so at the beginning, there's a book called Patent It Yourself, and you can do patenting successfully without an attorney. But you'll probably, if you're positioned the way I think most of us in the room are, you'll probably go through your tech transfer office. And what they do is they have attorneys for different kinds of things, right? I mean, Baylor Med, for example, probably only has people with medicine and biology. U of H, we have, we invented high temperature superconductors, we have ceramics experts, and we have et cetera, et cetera. And so the tech transfer office has different outside attorneys they use who specialize in particular areas. But I don't have a good answer to your direct question, which is if you as a non-lawyer inventor want to do the best job you can in patenting in life sciences, is there a written thing? The big things to know about, just Google the following: the Amgen decision and the Myriad decision. Those are two great big important court decisions that affected patenting in this space. And Amgen is fairly recent, and it narrows the kinds of things that can be patented successfully. It's the one that says you can't patent all anti-insulin antibodies. I'm exaggerating. That's an exaggeration of the truth. But it's harder. So that one militates, depending on what you're working on, we're doing a lot of sequence listings, which is why our recent provisional is 130 pages long, because we just listed the heck out of it, and a lot of derivatives there. Oh, by the way, while I was doing that, we started a reasonably successful company based on glow sticks and another one based on glowing objects like this. And I won't have a chance to talk about it, but I want you to, that works pretty well. I want you to open the pregnancy test and use it for whatever you want, but take it apart and look inside. And especially notice the cassette. And the black object is a cassette that we have injection molded for our startup in Hyderabad, India, in large numbers. And it's highly engineered. It's not as trivial as you would think. And the glow stick is because one of our things was based on glow in the dark materials and the other one was using glow sticks. Pretty good. But anyway, there's a question about it.
Thanks for your talk. Just a quick question about the timing of startup. So for normal medical or pharmaceutical products, to take very long-term development before they go to market, like phase 1, phase 2, phase 3, and taking a very long time to get FDA approval. So what will be a recommendation for good startup timing? Well, after I accumulate some data effective, and it's time I can start up like ICOP or small business fund from NSF, or I should go to the middle point of a product development, or I were very close to be commercialization.
So I would start early, in part because, especially with a therapeutic, you want to nail the IP down very carefully. You really, really don't want to put large amount of efforts into a situation where you haven't got the IP 100% nailed down and probably filed internationally. And that means you need money early because international filings cost money, et cetera, right? And so, especially in therapeutics, I would tend to start. Because you're not going to get, unless you're personally wealthy or have interesting friends, you're not going to get through to phase 2 trials without having incorporated a company, let people buy equity or get some formal legal arrangements. You know, when we sold this company that I referred to that Jim Briggs was prominent in, you know, the paperwork was about that. There's a lot of stuff happens, a lot of paying of lawyers and things like that. But I think if you're doing therapeutic and you're serious about it, you're gonna start early and you need to raise money.
Since you have already sparked my curiosity, so what is glow stick chemistry, if you would like to talk a bit more about it?
So I was eager to talk about it. The short summary is that lateral flow is a fabulous technology, but sometimes it lacks analytical sensitivity, so you can't see pathogens, or it lacks quantitation, so you can't do biomarker things. And so we developed 2 ways of making the particles glow in the dark. One is with these phosphors, you know, this is a strontium aluminate that's glowing pretty well. And also the glow sticks thing that I handed out, these have 2 chemicals in them plus a fluorescent molecule. When the 2 chemicals are blended together, they make a reactive intermediate, which is a cyclic dimer of carbon dioxide, the world's most unhappy molecule. But when it bumps into the fluorescent molecule, it excites it chemically, no optics, which means I can get fluorescent levels of sensitivity from cheap self-stable liquids instead of fancy optics with interference filters and the emission, excitation, and cubes and all that kind of stuff. I mean, all I have is this, and one of our tests uses, you know, 2 cents worth of these liquids, and they're shelf stable for years. So that makes a pretty good, you know, this will be in the PDF. But with the glow in the dark objects, we have a pretty promising home test for lupus nephritis, which is a nasty, dangerous complication, normally now tested by sticking a needle into somebody's kidney and taking out a piece of meat. Patients will do that 1 time. And with a brilliant rheumatologist collaborator, Chandra Mohan, who discovered great biomarkers, we have a urine test that will probably work. So I don't know if it will use our glowing object platform. I don't know if it will use the one marker that we focused on. There are others. But the idea that, you know, in 5 years, people with lupus will test themselves for nephritis, I'm pretty comfortable with that. And then for glow sticks, we did this. This is the chemistry. It'll be in the PDF. And it was the 2nd most sensitive lateral flow for COVID in the NIH RADx program. And we asked who was number 1. And number 1 was our spinoff, 2 of my grad students using this thing. We had a lot of fun during the COVID time. I know we suffered a lot.
Any questions?
So you did mention that patient with lupus nephritis may be able to like test himself using this technology. Do you think along the way other diagnostics could be made easier using the same pattern? Like
Yes.
in general, other nephritis?
Yes. You said other nephritis?
Yes, other types of.
Yeah. So, I mean, I think it's a very good platform for diagnostics in general. This, I won't go into the details, I'm conscious of time, but the categories here are healthy controls, lupus but inactive, other kidney diseases is the ANR, the green ones, and then active lupus nephritis is the red ones, right? So you'd want other markers. The whole point of this marker is it doesn't respond to other kidney diseases. But sure, and it's a great platform for all kinds of other stuff. The problem is that we ended up, I mean, I had an interest in one company and we started another company to do COVID diagnostics, whereas COVID was a once in a century event that went up, down, right? And so diagnostics companies focusing on COVID just got wiped out. But we still have the platform technologies and we're using it for other things now. We have a couple of grants for the glow stick thing.
Okay, we've got 2 questions from chat. First one is, can you comment on the importance of a patent portfolio surrounding the core invention as compared to the initial seminal patent?
Yeah, that's a great question. It's one of the things you need the money for, right? So for example, the original glowing object patent for lateral flow was developed internally at U of H, and I'm an inventor on that. But the spinoff, which went out and raised 28 million non-dilutive dollars and got to 120 employees, did a bunch of other stuff too. Luminostics, now called Clip Health. And they had their own IP, fleshing out the whole story. And that's a very valuable part. But notice that assumes you can get a bunch of money and do things. So the fundraising part is, I have sold a single bare patent for a lot of money, but it's much better if you have a whole portfolio.
Thank you. Okay, second question. Is a patents necessary to launch a startup company?
No, I mean, there are many, many companies that get started without any patents at all. I don't know, for example, if Uber has any business method patents. Those kind of went out of fashion. There's a restaurant near me I like very much. They're doing really well. They have no patents. Sometimes, first mover advantage. I mean, there was a thing called a pet rock. The pet rock sold rocks, but it was cute and they had little birth certificates and they packaged them really well. No patents, but for a period of about 5 months, there was an insane craze. Then there was the sand breeding kit. So you could have cute little baby sands, right? And people gave a lot of money to these people. Somebody retired a multimillionaire on this thing. No patents, just a certain amount of creativity and chutzpah. It's I'm going to sell rocks now, right? So there are plenty of ways to make money. But in our areas we're mostly talking about here, science, technology, medicine, pharmaceutical development and stuff, patents are really important. It's usual.
Any more questions? Yes, sir.
Thank you for all that. I'm wondering about the authorship, how to decide who is the author and what decides who is not the author.
Right, and we're going to talk specifically about patents and inventors on them, right? Yeah, okay. So I took that slide out for length. Someone who acts only under the direction of somebody else is not an inventor and shouldn't be listed. So there's a tendency to want to include your friends, and you should be careful about that. One thing you can do is invite someone to become an inventor. Like, please, I have this. I'm sure with your brain, you can think of some more stuff in this space. We'll put it in the patent. Then you'll be a legitimate inventor. But don't just assign arbitrary inventorship. Arbitrary authorship, we could argue, honorary authorship, we could argue about for papers. But for patents, it can blow you up. On the other hand, be careful of leaving people off because they may come and sue you later if there's an awful lot of money. And we've seen many examples of that. Send me an email, or if you just Google, there are pretty good discussions about this. If somebody has to have made a real intellectual contribution to what turns out to be an issued claim at the end, not even the filed stuff. That last part, the issued claim part, is often.
I have a question on the patent modification or patent portfolio. So when you license it out to a company, and when the company modifies or builds a portfolio around it, what's the role of the university's tech transfer office into that process? Do they have any role, or are they completely out of the loop when they develop that process?
I once called somebody up and said, "You owe me $250,000." And he said, "Strict reading of the contract." I said, "Oh yeah, you're right." And I shut up and I came back like 8 months later and got my money. So you want to have good contracts here. And a thing that will commonly be in such a licensing contract is that if somebody makes an improvement, the originator is allowed to use it under some terms. You can negotiate any terms you want, but people will think about that case because you don't want to get frozen.
All right, one more question from chat. Could you comment on the primary challenges and future prospects of antigen detection lateral flow assays?
So all challenges will be resolved by my fantastic technologies for antigen detection lateral flow assays. Well, frankly, one problem is it's not a tremendously profitable industry in most cases. One is that a zillion companies rose up during COVID to try to make it better in various ways. We're still doing pretty well competitively. We're still getting grant money and we still have good sensitivity. One is that we want it to be simpler because everybody wants it to be done at home, over-the-counter stuff, right? I mean, we have a grant from NIH right now to try to do HIV detection in rebound. That is, you get infected with HIV, one of the first signs is seroconversion, right? You raise antibodies against the virus. And that's a massive amplification of signal. You know, zeptograms, picograms of virus produces milligrams of antibody in your blood. Easy to see the antibodies, you're seroconverted, you've been exposed to HIV. Fine. Then you take your drugs, the modern drugs are great. They knock you down to non-detectable level, you're fine. And then for some people, one of two things happens. They get a mutation, the virus comes back, or they're, you know, living rough in a variety of ways, substance abuse, et cetera, and they don't take their meds and the virus comes back. Now you've got a problem where the virus has come back, still at a really low level, they're still antibody positive. So you can't look at seroconversion anymore. You have to look for the virus. Well, we have a way of doing that, you know, RT-PCR, but that's a big fancy machine not available at home or in clinics or shelters, et cetera. And NIH is very interested in having much simpler ways of doing that. Complicating it, the virus and the viral proteins are covered with the antibodies that the host has already raised. So your diagnostic antibodies can't get in there. So you have to do a thing called immune complex association too. So we have a grant for that, right? So, you know, the simple things like pregnancy tests are done. So one of the challenges is we're going for harder and harder targets. But I will note that the advance of microelectronics and DNA sequencing, et cetera, really provide tremendous support for diagnostics.
Any further questions from anyone or in chat? All right, well, let's offer Dr. Willson a thank you.
Yeah.